Howdy, my name is Alondra Curiel, and this is our hand washing experiment.
Howdy, my name is Cassandra Fernandez, and I hope you'll enjoy. Introduction. What is the purpose of this experiment? The purpose of this experiment is to compare how effective water stands versus soap and water are at reducing microbes on the thumb over different variations of washing times. Hand hygiene reduces the spread of microbes on the skin, some of which can cause disease. Blood agar supports microbial growth and shows changes in bacteria.
Materials and methods. In this lab, we used four blood agar petri dishes, distilled water, soapy sponges, and 8 cotton swabs, as well as saline.
The procedure was started off with the right thumb, with that contains only water. The first step we went through was labeling two blood agar plates labeled R1, R2, R3, R4. We chose one individual from the group to complete the hand washing portion, while the other swabs and inoculates the plates. The right thumb was used with only water. R1 was swabbed before washing, then inoculated in the corresponding plate labeled R1. R2 washed under distilled water for 1 minute, then swabbed and inoculated in the corresponding plate labeled R2. R3 was washed for a total of 3 minutes, then swabbed and inoculated in the corresponding plate labeled R3. R4 was washed for a total of 6 minutes, then swabbed and inoculated in its corresponding plate R4.
Now for the left thumb. Um, Two other blood agar plates were individually labeled L1, L2, L3, and L4, each labeled with soap. The left thumb was used with water and soap. So L1 was swabbed before washing and inoculated to L1. L2 was scrubbed with soapy sponge for 1 minute and it was rinsed with distilled water, then swabbed again to be inoculated in its respective plate. L3 was scrubbed with a soapy sponge for 3 minutes, rinsed with distilled water, then swabbed and inoculated to its respective plate as well. Same thing with L4. It was scrubbed with a soapy sponge for 6 minutes and rinsed with distilled water, then swabbed and inoculated.
Results: right thumb with distilled water, R1 through R4. R1 and R2 contain no bacteria seen on R1. Very slight bacteria on R2 and hemolysis can be seen on some of the bacteria growth. R3 and R4. There is no bacteria present on either side. This could be due to human error, like not wiping thumbs with enough pressure, or just a case of really clean thumbs.
Results for the left thumb with soapy sponge, L1 to L4. For L1 and L2, more bacteria growth than the right thumb is present, and hemolysis can be seen in some of the bacteria growth here as well. And L3 and L4, L3 does not have any bacteria, and L4 does have a bit, which is a bit odd because L4 was washed longer with soap and water than L3 was. And on this plate, we can also see some hemolysis present as well.
Conclusion: the hand washing experiment demonstrated that rinsing with only water was ineffective at reducing microbial growth, as evidenced by the numerous colonies on R1 through R4. Washing with soap significantly reduced the overall number of colonies L1 through L4, showing that soap is more effective at removing bacteria. However, hemolytic colonies were observed on the soap wash plates, indicating that while soap decreases microbial load, some affect potentially pathogenic organisms such as hemolytic bacteria may still persist. These findings emphasize the importance of proper hand hygiene practices, as though washing with soap and water is essential for minimizing both the quantity and potential pathogenicity of microorganisms organisms on the skin.
Thank you very much for watching our lab video.